Aquatic invasive species have the potential to cause serious ecological and economic damage in Minnesota. Researchers at the University of Minnesota's Aquatic Invasive Research Center are busy conducting important studies on how to combat the spread of starry stonewort, zebra mussels, and carp. First, we're going to talk with Dan Larkin and his team as they explore ways to effectively grow starry stonewort under laboratory conditions.
Today we're going to be looking at an aquatic invasive plant called starry stonewort that was first found in Minnesota in summer 2015. Right now in these different tanks and buckets and aquaria, we are growing starry stonewort that we've collected mostly from Lake Coronis as well as from Upper Red Lake. And so what we're trying to do is get a good handle on how to keep it alive and healthy and reproducing, so that when we do experiments to look at desiccation or herbicide treatments, we have good healthy plant material to work with. This is starry stonewort that we collected from Upper Red Lake. You can see it has a nice green color, thin stems. It's been described as sort of grass-like. People are concerned about having starry stonewort in our lakes because it can grow so densely in certain areas. It can spread and cover large portions of the shallow water along lakes' edges and can also fill quite a bit of the water column. Unlike some of our native charophytes that stay pretty low down to the sediment, starry stonewort can grow up higher and start to fill in those shallower waters. So starry stonewort is called starry stonewort because it has these star-shaped structures called bulbils. They look like little white stars, and those can give rise to new plants. So this is a bulbil, one of those star-shaped structures that is now sprouted and is giving rise to a new plant. You can see that green stem that's grown out of it. The most likely way for starry stonewort to be spread is by boaters. Where it's been found so far has tended to be concentrated near accesses and in lakes that have accesses. It's not producing spores to allow for sexual reproduction here in North America. The spread is by material being transported and creating clonal or vegetative spread. So it's very unlikely that waterfowl or other wildlife are spreading starry stonewort. It's likely to be driven almost entirely by boater movement and other human activity. I think the real key to make sure you're not spreading starry stonewort is to closely examine your boat, your prop, your trailer, looking for the green plant fragments, and just, a relatively small fragment can lead to a whole new plant. And then also looking out for these white bulbils that sort of might be hidden on things that have a white background. For example, if it's stuck to the hull of your boat, it might not be that obvious trying to find them. So really carefully inspecting and cleaning boats and other equipment that you're using out on the water is a key to preventing further spread.
Next, we join Sunil Moore to learn about his research into pathogens to control invasive carp.
My role in this project is we are working on screening the pathogens that are present in invasive carp species, mainly the common carp, silver carp, bighead carp. And then next phase will be to figure out the potential pathogens that we can use to kill the carp in different lakes.
Sunil grows carp cells and inoculates them with viruses. They are checked daily for 7 to 10 days to see if any of the viruses are growing. The object is to identify pathogens that could kill invasive carp without harming native fish or other species.
Once we know all this, then it will be easy for us to pick up a potential pathogens that can kill carp only, but will not affect our native population.
And finally, we're in the laboratory with Adam Dahl, who's doing research on zebra mussel larvae.
The research that I'm doing is on zebra mussel transport through recreational watercraft. So there's been lots of studies and there's lots of information out from state agencies across the country about moving adult zebra mussels from place to place, and there are laws in Minnesota about draining water and making sure you're removing everything. So we're looking at what's next and what's left. So we're trying to look at the last little bits of water. We're calling it residual water in watercraft. So it's very small amounts of water that are potentially being moved from lake to lake. So we're collecting those as samples and taking a look at them under a scope. And currently we're just getting counts of what we're finding in there of the baby zebra mussels. They're called zebra mussel veligers. It's the larval stage. It's free floating in water. They're invisible to the naked eye. So they could easily be transported in this very small residual water. And we're focusing at two very busy lakes, Lake Minnetonka here in the metro area, and then we're also looking at Gull Lake up near Brainerd. First, we have to collect samples at the access, and that's done after the boater has done everything that they're legally required to do. So they've drained their live wells, bilge areas, everything like that. And we go in with a syringe and suck up the last little bit of water. We measure it out, and then we preserve it in an ethanol sample, and we get a bottle like this with a sample. And then what we're going to do is filter it through some fine mesh and then put it into a petri dish and look at it under a microscope. So now that we've concentrated the sample, what we'll want to do is take it over and look at it under the microscope to see if there's anything present. When we're counting and looking for zebra mussel veligers, we're actually using a polarizer to look at the sample under polarized light. And you can see the whole background turns black, and there are a few white blobs on there with black X's on them. This is a zebra mussel veliger right here. This is a zebra mussel veliger. So there's one right there too that's a little smaller. So there's 5 in our view screen right now. So under the polarized light, the shell of the zebra mussel is essentially glowing, and there's a little X on them, and that's what we're counting inside the sample. A recommended option that we have here to go above and beyond would be to dry your watercraft. Minnesota recommends at least 5 days to make sure that all of that residual water dries out. And you could also look for a decontamination unit that the state stations all across the state and have your watercraft decontaminated with hot water, which is scientifically proven to kill adult and veliger zebra mussels.
This segment has been brought to you by the Aquatic Invasive Species Task Forces of Big Stone, Candy, Ojai, and Yellow Medicine counties.